Welcome back to another video, guys. Today we're going to be talking about carbohydrate digestion and absorption. And if you guys benefit from the video, please go ahead and leave a like, comment if you got any questions, and please subscribe because these videos takes a long time, guys. Okay, so let's get started. So before we dig into the digestion and absorption stuff, we have to talk about a little bit about the types of carbohydrates we have, okay? So here we got carbohydrates, okay? And you can actually split this carbohydrate into simple carbs, or you can do some complex carbohydrates, okay? So what would be your simple carbohydrates? Or your simple carbohydrates, your simple sugars, they're going to be your monosaccharides. Let's write that here. So they're going to be monosaccharides, and they're going to be some disaccharides, okay? Your complex carbs, well, they're going to include some things like oligosaccharides and polysaccharides. Okay? So again, so far we've had these carbohydrates. You can have simple or complex carbs. The simple are the monosaccharides and the disaccharides. So the monosaccharides, these include the simplest forms of sugars you can actually have. And there's about 4 of them. Okay, so one being glucose. Everybody knows glucose. The next one is fructose, which is a natural sweetener here. And we also have galactose. And then finally, one more is ribose, which we actually use for DNA. Okay, so those are your monosaccharides. What about your disaccharides? Your disaccharides include things like sucrose, which, by the way, is made up of glucose and fructose, and lactose, which is glucose and galactose. Okay. And then finally, we have maltose. And again, maltose is 2 glucose units, okay? So a disaccharide would look something like this, okay? And we'll see more of it later, but it would look something like this, okay? Boom, 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 bonded to another monosaccharide. So this is one monosaccharide, this is another monosaccharide, and together they're called a disaccharide, okay? Okay. And these are the types of disaccharides. We'll come across them in today's video. Now, with regards to oligosaccharides, there's a couple that I said I would want you to know for human nutrition. They would be stachyose, verbicose, and raffinose. Okay. Now, all of these kind of differ in their structure. So stachyose, for example, is a tetrasaccharide. Verbicose is a pentasaccharide. And raffinose is a trisaccharide. Okay? But oligosaccharides are generally classified as anywhere between 3 to 10 sugar units. Okay, so sugar units. So for example, anywhere between 3 to 10 glucose units would classify here. And again, you have other polysaccharides, for example, oligosaccharides. So you can add in like maltotriose here if you want, right? Maltotriose is 3 glucose units. And then within this complex, you have other stuff here like polysaccharides. Polysaccharides are generally said to be greater than 10 sugar units, okay? So 10 sugar units, okay? Or 10 monosaccharides if you want, greater than 10 monosaccharides. And you can classify multiple compounds here, okay? So, for example, you can classify starch and glycogen, okay? And glycogen. You guys have heard of glycogen, I'm sure. Mostly found in animals. It's a main storage form of glucose in our bodies. But starch, if I actually break the starch down a bit more, actually let me do it like this. So glycogen. But starch can actually be made up of these 2 components. One is amylose, and the other one is amylopectin. Now, I'll show you the structure for amylose, amylopectin, and glycogen next slide, or at least a rough estimate of these molecules. But technically, these are all polysaccharides. Now, you can actually classify a bit more. So you can go into fibers, and you can start talking about glycosaminoglycans. So you got fibers, and you can actually split fibers, again, into things like cellulose, pectin, and hemicellulose. Okay. And obviously, these guys are going to be different types of fibers. So you can have insoluble and soluble fibers. Okay. So hemicellulose more often than not is an insoluble fiber. And then, of course, you can also have things like glycosaminoglycans. So glycosaminoglycans. And, that's going to be stuff like chondroitin sulfate and hyaluronic acid. Chondroitin sulfate, you find a lot of it in your joints. Chondroitin sulfate and hyaluronic acid. Okay, but again, I just kind of wanted to show you that this is what carbohydrates is. Okay, so when we talk about carbohydrates, we're talking about simple carbs or complex carbs. Simple is going to be some monosaccharides and disaccharides. Complex is going to be oligosaccharides and polysaccharides. Now, we're really mostly going to be dealing with this stuff, and this stuff. Okay, as well as these. So these oligosaccharides, by the way, important to mention that things like stachyose, verbicose, and raffinose, they cannot be digested by human enzymes. But, for example, in the gut microbiome, we have bacteria there that can actually digest these. And these, oligosaccharides are usually present in things like beans, peas, and bran, and they can actually cause flatulence. Okay, so once they're digested by the colon bacteria, they can cause flatulence. So these three. Okay, so that's a little bit about the carbohydrates, and now let's kind of go ahead and look at some structures here. Okay, so before I actually get into here, okay, this is going to be the structures of amylose, amylopectin, and glycogen. I want to talk a little bit about glucose, galactose, and fructose, which I said these are like your three main monosaccharides. So when it comes to all three of these guys, they're all 6-carbon sugars. Okay, so how do you know this is 6-carbon sugar? Well, you can start counting, right? But there's a specific place where you start counting. You start counting at this carbon here. So you start number 1, then 2, then 3, then 4, then 5, and then 6. Okay, so that's 6 carbons. And it's the same thing for this other monosaccharide, galactose. So 1, 2, 3, 4, 5, 6. Now, for fructose, you do something a little bit different. You actually start counting over here. You start going 1, then 2, then 3, then 4, then 5, and then finally 6, which is that carbon over there. Okay, so they're all 6-carbon sugars, but you can see that fructose is a little bit different than these other two. Now, galactose and glucose, if you guys have taken chemistry before, they exhibit a chirality between them, meaning they have a diastereoisomerism. Okay, what this means is that I can actually take this guy and this guy, but I can't superimpose them on one another. Okay, so if I try to plop this galactose down on the glucose, you can tell that they're not the same molecule. Okay, it's kind of like trying to impose your left hand on your right hand, right? They're not going to match. Okay, while they look the same and they have the same chemical formula, they're not going to impose on one another. So what is the actual difference between them? Well, on the 4th carbon here, the hydroxyl group is pointing up, and on the 4th carbon here, the hydroxyl group is pointing down. That is the only difference between glucose and galactose. That's it. That's the only difference between them. Okay. Now, if you look at fructose, that's a completely different than both of these guys. Okay. All right. Now, another term I want to introduce to some people who maybe haven't taken chemistry before is the term anomeric carbon. So the anomeric carbon here is this first carbon, and it's because it's doubly bounded to an oxygen. Okay, so it's bound to this oxygen here and this oxygen here. Okay, and it's a carbonyl carbon. A carbonyl carbon is usually written as something like this. Okay, so there's a carbon doubly bounded to an oxygen, and one R group here and another R group. The R group is just some sort of carbons attached to it. Okay, it doesn't matter. But the point is, this guy is known as an anomeric carbon. And the position of the hydroxyl group on this anomeric carbon is very important. Okay, and I'll talk about that a little bit later. And again, for galactose, the anomeric carbon is here because it's doubly bounded to this oxygen and this oxygen. But if you look at something interesting here, on fructose, the anomeric carbon is not on the first carbon, it's actually on the second carbon, okay? And, again, I'm not going to go into it, but this was actually called a hemiketal. This one's called a hemiacetal. Yeah, this is not a chemistry course, so I'm not going to go into it. But this suffices for you to know the sort of general structure of glucose, galactose, and fructose, and that this first carbon here is the anomeric carbon, which means that it's just doubly bounded to an oxygen group, okay? And the only difference between glucose and galactose is the position of the hydroxyl group in the 4-carbon. Okay, cool. That's that. Now let's go ahead and look at some of these different molecules. So remember, if I go back a slide, I said that polysaccharides include starch, and starch is actually made up of amylose and amylopectin. And we also have another kind of polysaccharide called glycogen, which is a storage form of glucose. Okay. So this first guy is a linear molecule, okay, meaning there's no like branches all over the place. It's linear, it's straight. This guy is known as amylose. Okay, this guy's known as amylose. And the one below it, you can see that it has these little red attachments. What are these red attachments? Well, these red attachments, they're a special kind of bond, okay? We call this red bond here, we call it an alpha 1 to 6 glycosidic bond. Glycosidic bond. So this is an alpha 1 to 6 glycosidic bond. And this specific bond here allows for a branching of the molecule. Notice how this guy didn't have any of those red bonds, so he wasn't very branched, okay? This guy has that branching bond, and he's able to branch the molecule out. What is this molecule called? This is called amylopectin. Okay? This is also found in starch. So both of these guys are found in starch. Okay? And, you can find these in cereal grains, potatoes, and other kind of vegetables. They're all going to be rich in amylose and amylopectin. Okay? Now, the specific percentages, for example, amylose contributes to about 15 to 20% of the starch, whereas amylopectin contributes to about 80 to 85% in the starch, okay? Okay, cool. Now, so we talked about these branched bonds in red, right? What about the regular bonds here that are just connecting these monosaccharide units together? Well, these regular bonds, these are going to be called, and let me do it here actually, these are called alpha 1 to 4 glycosidic bond. And they just allow for a linear chain of molecule, okay? So you can see there's alpha 1,4 bonds here, but there's also alpha 1,6. So amylopectin, not only does it have alpha 1,6 bonds, it also has alpha 1,4 bonds, whereas amylose only has alpha 1,4. That's why it's a straight linear molecule, okay? We have another molecule here, and this guy's named glycogen. And glycogen is usually found in animals, okay? You find none of it almost in plants, but it's found in animals, and it's a storage form of glucose for us. Glycogen, I want you to remember, glycogen is more branched. It has way more branches than amylose. Sorry, than amylopectin. Okay? So glycogen has more branches than amylopectin, and you can see that here, right? So branch here and here that we didn't have before, okay? Now, Again, remember, all of these branches are possible only because of this red bond here, which was called an alpha 1,6 bond. Cool. But what does this bond actually kind of look like? Well, just to help you understand it a little bit more, let's go back to this tiny bit of chemistry. So here we have a glucose molecule, okay? And here we have another glucose molecule. Remember I said there are carbons here, so carbon 1, 2, 3, 4, 5, 6, okay? This first carbon, we called it an anomeric carbon. And I said the anomeric carbon has a hydroxyl group attached to it. Remember? There's that hydroxyl group here. The position of that hydroxyl group is going to determine what we call this bond. So if the hydroxyl group is pointing down, as it is in this scenario, we actually call this bond, we call it an alpha 1,4 glycosidic bond, okay? And it's when the hydroxyl group here is actually pointing down, or you know, if you're looking at a Fisher projection, right, okay? So down and right is going to be this alpha 1,4, okay? Now, what if it isn't pointing up, down? What if it's pointing up? Well, if the hydroxyl group is pointing up on the anomeric carbon, then you're going to have this weird kind of bond now, okay, between another glucose molecule. What's that other kind of bond called? Okay, this bond here is called a beta 1,4, okay? Beta 1,4 glycosidic bond. And the reason for it is that, actually, let me move this up. So this is a beta 1,4 bond, and the reason is because the hydroxyl group on the anomeric carbon is pointing up. And again, you might sometimes see it written as left. Okay, so up and left. Okay. Now, what do I mean when I say 1 and 4, and what do I mean when I say this? So we talked about why it's alpha and why it's beta, right? That's because of the hydroxyl group. Why is it 1 and 4 and 1 and 6? Well, the 1 and 4 refers to the carbons. So here's the first carbon of this glucose molecule binding to the fourth carbon of this other glucose molecule. How do I know that? Well, you start from the oxygen, right? You start counting from the oxygen. Here's the 1 carbon, 2 carbon, 3 carbon. Okay, 4 carbon, and 5 carbon here, and I haven't drawn it, but there would be a CH2OH here, which would make this the 6 carbon, okay? So 1 and 4, okay? What about here? Here we go, 1 carbon, 2 carbon, 3 carbon, 4 carbon, okay? And there's a 1 carbon here, so this is a 1 and 4 bond. Cool, pretty easy. What about the other bond? This other red bond that I said was responsible for branching points. Well, that bond is going to be this guy right here. So let's start counting from the oxygen. 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbon, and then 6 carbon. Okay? So we have the first carbon here, and the hydroxyl group is pointing down. So if it's pointing down, we know it's going to be an alpha bond. Let's write that down. So it's alpha bond. And that's 1. So 1 carbon. And then let's count from the oxygen again. So 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbon, and then 6 carbon. Okay, so it's this 1 hydroxyl is attached to the 6 here. So that's an alpha 1,6 bond. Okay? That's how we get these. That's how we get these, and that's what there's a branch point. Okay? So just to make it clear, this alpha 1,6 is for branching, and the alpha 1,4 is for maintaining this sort of linear structure. Okay. So now in terms of digesting all of these large polysaccharides, because you know when we don't eat simple carbs more often than not, right? We're eating oligosaccharides and polysaccharides, okay? So when I'm eating these polysaccharides, how do I digest them? So you know there is a specific enzyme. This enzyme here is going to be called, and let's draw it in this color. This is actually called amylase, okay? Now there's different kinds. There's salivary amylase, there's pancreatic amylase. We'll talk about it. But for example, this alpha amylase, it is very specific for cutting alpha 1,4 bonds. So it's going to come here, 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 and here, okay? And it's going to cleave every single one of these bonds, okay? It's going to cleave all these bonds. Great. That's going to give us monosaccharides. That's what we want, right? Because when we're digesting and absorbing, we can't actually absorb large polysaccharides. We can only absorb monosaccharides like galactose, like glucose, like fructose. Okay? So that's what we want. Okay? So how do we do that? We have these enzymes come and cleave these alpha-1,4 bonds. Okay. But what about the alpha-1,6? Can it cleave the alpha-1,6? Well, the answer is no. Alpha amylase has no specificity for alpha-1,6 bonds. So it can come here and cleave this bond, okay? But it can't cleave this red bond here or this one here. And another fact is that alpha amylase is not very good at cleaving alpha-1,4 bonds that are adjacent to alpha-1,6 bonds. So it's not going to be very good at cutting this bond, or this, and this. Okay? So in this scenario, you might only end up with one monosaccharide unit freed. So you can see the problem here is we need to get rid of these alpha-1,6 bonds, and we'll talk about how later. Same thing over here in this glycogen molecule, okay? Within this glycogen molecule, I can maybe clean out two of these bonds. Okay, so maybe this one and this one, okay? But for example, I wouldn't be able to do these or any of these, okay? All right, so that's that. Now, just wanted to give an intro on how amylase works, and now let's kind of go into a deeper look at what's happening at the level of the oral cavity, what's happening at the level of the stomach, what's happening at the level of the small intestine, okay? So let's go ahead and dig right in. Okay, so at the level of the oral cavity, okay, you're going to have ingested two things here, okay? So first you ingested amylose, which was that linear molecule, and then you ingested amylopectin, which was that branched molecule, okay? Now, remember branches, amylopectin, it had alpha-1,6 bonds, so we can't cut that. At the level of the oral cavity here, we have a specific enzyme being secreted within the saliva. And that enzyme, if you guys recall from the gastric video we talked about, it was called salivary alpha amylase. So salivary alpha amylase will go and act on the amylose and the amylopectin, and just like what we showed in the last slide, it would do exactly this. It would do exactly this, so it would be able to cut out some of the amylose, it'll be able to come out some of the amylopectin, okay? And you might be asking, hey Keon, why didn't you mention glycogen when we're eating? Because even when you're eating meats, there is such a trace amount of glycogen in there that it's just not worth kind of mentioning it. The main kind of carbohydrates here are going to be these amylose and these amylopectins, okay? And we're going to look at them. Okay, so with this amylose and these amylopectins, salivary alpha amylase acts on them and creates these molecules. These molecules that are left over of the digestion of these guys are called dextrins. Dextrins. But specifically, this one, that was a remainder of the amylopectin, is called a dextrin with alpha-1,6 bonds. And this one is a dextrin with no alpha-1,6 bonds, okay? So it's got alpha-1,4 bonds, okay? With alpha-1,4. So you had amylose and amylopectin, right? And salivary alpha amylase started acting on them and broke them down into these molecules called dextrins. And the amylose ones were with alpha-1,4 bonds, but no alpha-1,6. And the ones that come from amylopectin are the ones with alpha-1,6 bonds, okay? Briefly, if you guys want to know, this is kind of the structure of a dextrin molecule, okay? It's just a whole bunch of these glucose molecules, monosaccharide molecules attached to one another, okay? And occasionally there will be branch points, okay? So, for example, this is what the dextrin with an alpha-1,6 would look like. And this is what a dextrin with an alpha-1,4 would look like. Okay? All right. But the whole point is we broke down some of those molecules. Now here's the thing. I don't know about you guys, but food doesn't stay in my mouth for a long time. I swallow that sucker right away. Okay? So very little of this salivary alpha amylase action is actually occurring here. So where does food go next? Well, remember we talked about it. You swallow the food and then it goes down through the esophagus and then it ends up in the stomach. So question is there any digestion in the stomach? Well, yes and no. Salivary alpha amylase, if you remember, I told you works best at a more alkaline pH. And we know that the stomach is very acidic. Okay, so the stomach has a low pH. So salivary alpha amylase will continue to break down these amylopectins and amylose until the pH of the stomach gets too low and inactivates alpha amylase. Okay, it inactivates the salivary alpha amylase, and at that point, there's no digestion. Okay, there's no digestion, and more often than not, we just ignore digestion in the stomach and we say there's no digestion. Okay, so low pH, and that's low pH is going to inhibit the salivary alpha amylase. Alpha amylase. Now, you might be thinking, Keon, are we only making dextrins at the level of the oral cavity? And some monosaccharides and disaccharides are produced, but again, the majority of it is going to be these dextrin molecules, okay? Okay, so if no digestion occurs at the level of the stomach, then where does the bulk of digestion occur? Well, remember again in the gastric video, I mentioned that majority of all digestion actually occurs in the small intestine, okay? So at the level of this duodenum. Now, at the level of this duodenum, you guys remember we had the liver, we had the pancreas, and then we had the small intestine. Sorry about that. We had the gallbladder here. We had the pancreas here. We had the duodenum. And we had this little vein actually connecting the small intestine all the way to the liver. That was called the portal vein. Okay? So this is where the food is going to end up. This is where those polysaccharides are going to end up, those dextrin molecules. And if I kind of zoom in on this one section, this is what I would see. Okay? So you guys remember how I said the small intestine, it has these little things, right? These little folds, these circular folds that were called villi, right? They were villi, and on the villi, I said, were made up of these little cells called enterocytes, okay? And on top of those enterocytes were even smaller little wavy things called microvilli, okay? So we had the villi that's made up of enterocytes, and then on top of the enterocytes, there was the microvilli, okay? Go back to the other video and watch that if you didn't know. So when food enters the lumen of the small intestine, right? So this is the lumen, okay? The little cavity. So when food enters this lumen, what kind of food are we talking about? Well, we're talking about the food, the dextrin. So let's actually make this font a bit smaller. So when this dextrin comes in, the ones with the alpha-1,6, and then there's other dextrins that come in with an alpha-1,4, okay? So they're going to come here and they're going to encounter a very specific enzyme. Where's this enzyme coming from? This enzyme was actually secreted from the pancreatic acini. Again, go back to that video if you haven't watched it, my previous video. I talked about the specific kind of cells that actually make the enzymes, and they secrete them into the actual the duodenum, right? But the pancreatic alpha amylase, that's what I'm talking about. So the pancreatic amylase. And if you guys remember, there was a specific sphincter here. We called it the sphincter of Oddi, or the hepatopancreatic sphincter, and a specific hormone that we talked about called cholecystokinin was coming here and stimulating the relaxation of that sphincter, opening it up and allowing for pancreatic amylase to come in alongside other stuff like bicarbonate. Okay, and remember why we needed this bicarbonate, because the chyme that is coming in from the stomach was very acidic. Okay, and so in order to neutralize that chyme, we wanted to secrete some bicarbonate from the pancreas to make the pH higher here, okay, more alkaline. Why is that beneficial? Because amylases, they like to work at a more alkaline environment. That's why beforehand, salivary amylase couldn't work in the stomach because the pH was too low. So remember, we need bicarbonate. Okay, so what happens when these dextrins run into this alpha amylase? So when the dextrins with alpha 1,4 run into pancreatic amylase, they're going to turn into something known as maltose, and the dextrins are going to turn into some other stuff, okay? So one of them is going to turn into maltotriose, and we talked about that. We talked about maltotriose. Another one is going to be limit dextrins, okay? Limit dextrins. And the other one is going to be maltose, okay? So the dextrins with alpha 1,6 bonds will further turn into limit dextrins, okay? These guys are about 8 oligosaccharides, right? So, sorry, not 8 oligosaccharides, 8 monosaccharides in length. And the maltotriose is going to be 3 glucose units with alpha 1,4 bonds, and the maltose is going to be 2 glucose units with alpha 1,4 bonds, okay? Again, we'll see that more over here. So what's going to happen, basically? We made, as the end product of digestion here at the lumen with pancreatic amylase, we made maltose, limit dextrins, and maltotriose. Cool. These guys are not going to be digested any further by this pancreatic amylase. They need to go and get digested here on the enterocyte. You guys remember I called this portion of the enterocyte the brush border? The brush border. They need to go here and get hydrolyzed by brush border enzymes, okay? So when we're actually cleaving these bonds between monosaccharides or oligosaccharides, that's called hydrolyzation, because I'm adding water into the reaction, okay? All right. So that's going to be that. Now, what's going to happen next? What's going to happen next? Well, you're going to get all these different things here, okay? You're going to get maltose, okay? You're going to get lactose, okay? And you're going to get some other molecules here. Now, you might be thinking, okay, Keon, where did lactose come from? So, you know we had dextrins here, right? And at the start we had amylose and amylopectin, okay? But you know when I'm eating food, I'm not just eating that, right? I'm eating other sort of carbohydrates, and with it comes things like sucrose, lactose, okay? And all that other stuff, okay? So those guys are also going to go and get digested, okay? So for example, this molecule here, this is sucrose. This one is going to be something called isomaltose, okay? And we have one more here called trehalose, okay? Trehalose. So, as you notice, we have these little scissor things sitting on the brush border membrane. What are these scissors? These are enzymes. These are enzymes, guys, okay? And we also have specific transporters here sitting ready to absorb monosaccharides. Okay, so let's talk about the different kinds of molecules we have here. So firstly, we have sucrose, which is just a disaccharide of glucose and fructose. Then we have this isomaltose, okay? Isomaltose is literally just glucose and glucose, but you see the bond is a little bit different. The bond is actually an alpha 1,6 bond, okay? Remember this bond here? I said this is the branching one, the one that allows for branching to occur, and you can see this right here. It's not linear, it's branched. Okay, maltose. Maltose, if you remember, I said is just going to be a glucose and glucose, but this one just has alpha 1, 4. Okay, so it's linear molecule. Now, lactose. Lactose is a special bond that we talked about at the start of the video, and that's called beta, right? So beta 1, 4. Beta 1, 4. Remember I said this carbon here, the first carbon is an anomeric carbon, and if you look at it, the hydroxyl group is pointing up. So if it's pointing up, that's a beta bond, okay? And it's between the 1 carbon of this molecule and the 4 carbon of this molecule. Now, what is lactose made up of? It's made up of glucose and galactose. Okay? All right. Then finally, we have this other molecule called trehalose. Trehalose is also used as a sweetener, just like fructose. And trehalose is, again, made up of glucose and glucose. But it has a different bond. It's actually an alpha 1, 1 bond. Just like sucrose. Sucrose is another bond. Sucrose is alpha 1, 2. But you don't have to know these, okay? So you don't have to know that. Don't worry about that. Okay, now there's going to be specific enzymes in this brush border that is able to hydrolyze, right, digest all these different kind of molecules that we have here. So this one here is called sucrase. Sucrase. And it's going to be able to cut the bond that's present on the sucrose, releasing 1 glucose and 1 fructose. Okay? What about this green guy? This green guy, he's very special. Very special. It's called isomaltase. You might see him have another name as well. He might actually sometimes be called alpha dextrinase. Or you might also see him be called alpha 1,6 glucosidase. Okay? So, again, the reason why I'm saying that, I'm just going to delete these names because it takes too much space, but the reason why I'm saying this is because this guy is very special. He is the only enzyme that is capable of cleaving alpha 1,6 bonds. He is the only enzyme that's actually able to cleave this alpha 1,6 bond that we had problems with, right? We said we can cleave alpha 1,4, no problem. Pancreatic amylase can do that, salivary amylase can do that, but none of the enzymes that we talked about so far can do alpha 1,6 bonds. The only one that can is this guy, isomaltase. And another tidbit here is that technically sucrase and also isomaltase are complexed together, meaning they're literally connected to one another, okay? That's why I put them right next to one another here, okay? But sucrase is going to take stuff like sucrose and hydrolyze that bond between them and give us glucose and fructose. Isomaltase will take isomaltose and cleave that alpha 1,6 bond. It can also take any of the limit dextrins that we got, and I'll show that later. Okay? So it can take limit dextrins with alpha 1,6 bonds and cleave those as well, freeing them and giving us some glucose and glucose. Okay? What is this other guy here? What is this enzyme? This enzyme here is called maltase. That makes sense, right? Maltase takes maltose, okay? And you remember we also made maltotriose? Well, that maltotriose can also be hydrolyzed by maltase, okay? Because all maltotriose is literally just this. This is literally the shape of maltotriose. Boom. It's 3 glucose units with alpha 1,4 bonds. But maltose is 2 glucose units with alpha 1,4 bonds, okay? All right, and so this guy is able to cleave that, okay? Cleaves alpha 1,4 bonds, and it gives us glucose and glucose. Now this guy here, remember we said lactose has a special linkage, beta 1,4? There are other molecules that have these special linkages. For example, cellulose. Cellulose has a beta 1,4 linkage. But we don't actually have the enzyme cellulase, okay, that is specific for this bond. But we do have an enzyme here called lactase. And lactase is very specific for lactose's beta 1,4 bond, so it's able to cleave that, okay? It's called a stereospecificity. Okay, so lactase has a stereospecificity towards the beta-1,4 bond of lactose. But because we don't have an enzyme that cleaves cellulose, it's actually a fiber because it's not going to be digested and absorbed. Therefore, it'll actually be fermented by the bacteria in our colon. Okay, so it's a fiber. All right. Now, finally, we have this other guy, trehalose. This trehalose molecule here, it's going to be cut by an enzyme called trehalase. Trehalase, which again gives us some glucose and glucose, okay? With lactose, it gave us galactose and glucose, okay? So how does all this stuff get in? How does all this stuff get in? So now we talked about all the enzymes, but how does this stuff get in? Well, on the apical membrane and on the basolateral membrane, we have specific transporters. So hold on, where's the apical membrane? The apical membrane is the membrane located, it's pointing to the lumen, okay? So it's the lumen side, okay? So this portion is the apical membrane. This portion is the basolateral membrane, okay? So remember that. So the apical membrane is on the lumen side. The basolateral membrane is the side facing the blood. Okay? Facing the blood. So on the apical membrane, we got three transporters. We have GLUT5 and GLUT2, and we have this other one here. This guy is called SGLT type 1. SGLT stands for sodium dependent glucose transporter type 1. Okay, so sodium dependent glucose transporter type 1. And the reason why it's sodium dependent is because it uses sodium. Now also, this is actually a secondary active transport. So secondary active transport, meaning it uses ATP. Okay, so it's an active transport that uses ATP, and furthermore, it actually is a symporter. A symporter is defined as taking two molecules and transporting them within the same direction. So look, this molecule, this transporter, is able to take two sodiums, okay, and then it's going to be able to transport either a glucose molecule or a galactose molecule, okay? So either one, not both, either one, and it's going to transport in the same direction as the sodium. Now, the way this works is that the sodium will bind first to the transporter, then it'll increase the affinity for glucose or galactose, and then the glucose or galactose can bind and then get transported. So without sodium binding, glucose or galactose can't actually use this transporter, okay? But either way, this SGLT1 is very important, okay? It brings in the glucose or galactose into the enterocyte, okay? This is the enterocyte, okay? Cool. And then this sodium comes in here, and on the surface of every cell in your body, it'll get pushed out and then get exchanged by potassium, okay? Cool. Now, what about these other transporters? So this GLUT2, it's not very specific. It will take galactose and it will take glucose, and it will take both of them in, okay? And now, again, you get another way of taking glucose plus galactose into the cell, okay? Cool. This GLUT5, though, is very specific. It only takes fructose. Okay? So GLUT5 is very specific to fructose, okay? And that's how we bring fructose into the cell. Now, after these guys are inside of the enterocyte, they have to now go into the blood. So how do they do that? Well, they all come here, okay? And they all collectively are able to go through this GLUT2 transporter. They're all able to go through this GLUT2 transporter, which, remember, I told you is not specific, okay? It doesn't care what it lets in, all right? Okay, so now if I was to draw it in the blood, right, in that portal vein, now I have 3 molecules. Okay, I have a glucose, I have a galactose, and I have a fructose molecule. Okay, pretty cool. What happens to these guys? Well, glad you asked. These guys are going to continue up the portal vein. Okay, until they reach the liver. The liver has another transporter. That transporter is a GLUT2 transporter. Okay, it's able to take all of these guys. Okay, it's able to take all of these guys and bring them into the liver. Okay, why? Because the GLUT2 is not that specific. Okay, now before we talk about what happens to all of these monosaccharides that we absorbed, okay, I have to mention something. I have to mention something about the Km, okay, or this Michaelis constant, okay. So what the Km is, if you guys need a refresher, right? This is not a biochemistry course, so I'm not gonna discuss how you derive the Km or what it truly is, but the Km you can think about is an indicator of the affinity, okay, of this transporter to a specific molecule. The lower the Km, the higher the affinity, okay, and the lower the capacity. So what does this mean? Let's take GLUT5. So GLUT5 has actually a very low Km, okay? And what that means is that it has a high affinity for fructose, but it has a very low capacity for fructose, meaning it can get very saturated easily. And if a transporter is saturated, it can no longer be used to transport an object across the membrane. Okay? So it has a very high affinity for fructose. It's very specific to fructose. GLUT2, for example, if I take GLUT2, remember we said it's not very specific? That means that it has a high Km, meaning it has a low affinity for all of these different molecules, right? It's not very specific, but it has a high capacity, okay? So it requires a high number of these molecules, like glucose and galactose. And that should make sense to you, okay? This should make sense to you because after eating, the concentration of this galactose and glucose is going to be so much higher in this region, right? In the apical membrane, than it would be inside of the cell, right? And so these guys are just going to passively go down their concentration gradients into the actual cell, okay? So we had 2 modes of transport. We had an active transport, and we had a facilitated transport, right? Which is with these GLUT transporters, GLUT5 and GLUT2. Now, technically, there are actually 14 different GLUT transporters, but today we're only going to talk about 5, okay? We're only going to talk about 5. Okay. One more thing that I want to mention regarding this GLUT transporter is that this apical GLUT2 is actually regulated by insulin. Okay, so after a meal is done, okay, you guys know that when when glucose makes its way into the blood, right, insulin starts to spike up, okay, and I'll show that. That insulin will then come all the way back into this apical membrane, and it'll tell this enterocyte, and it'll say, hey, listen, you should probably get rid of this GLUT2 on this membrane now. Why? Because we already have glucose in the system, and I don't want you to absorb any more glucose, okay? So it'll actually inhibit this transporter. It'll inhibit this transporter, okay? Cool. What about the Km of this SGLT1? Is it high or low? Well, the SGLT1 is actually low, okay? It has a low Km, meaning what? Meaning it has to have a high affinity for these glucose or galactoses. Why? That makes sense. Because sodium, if you remember, I said has to bind here, and that increases the affinity for glucose and galactose. Okay, and it's also an active transport mechanism. Okay. All right. Now let's follow these monosaccharides into the liver and see what happens to them. So now that we've absorbed them and they have gone through the portal vein and now they're in the liver is the first organ to see them. The liver is the first organ to actually see them, okay? You know what the liver is going to do to them? The liver is going to get this glucose here. It's going to get some galactose here. And it's going to get some fructose, okay? So, briefly, I'm not going to go into the biochemistry of it, but the liver is going to take galactose and fructose into glucose, okay? So it'll take fructose and turn it into something called fructose 1-phosphate, okay? And through a series of enzymatic steps, it'll turn into glucose. Same thing with galactose. Galactose will turn into galactose 1-phosphate, and then eventually to glucose. Now the glucose that comes into the cell here, okay, it has a couple of paths it can take. So it comes here, okay. It can either be turned into glycogen, okay. It can be pushed to other pathways, okay. It can be pushed, for example, to energy production. So pyruvate, which then goes into the Krebs cycle or the TCA cycle, and then the electron transport chain to make ATP. Okay, so it can be used for this instance. The glucose can also be pushed out into another pathway called the pentose phosphate pathway. And the pentose phosphate pathway is basically what we use to make ribose sugars, so we can use them for nucleotide synthesis. And that starts with glucose, actually, okay? You can also use it to make fat. So that's called, making fat from glucose substrates is called de novo lipid synthesis. Okay, so you can see how this glucose can go into many different pathways, but what decides where it goes? Well, the majority of that glucose is actually going to spread out, okay? So the liver is actually going to let go of a lot of that glucose, okay? It's going to push a lot of that glucose out, okay? Where is it going to push it out to? It's going to push it out to here, 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 and here. All right. Now, before we go and follow all of these different organs and how they're kind of using the glucose, okay, I have to say something very important. First things first is that the liver is very efficient in its use of glucose, okay? So because it's the first organ to see glucose, it's not going to hog all of that, okay? It's going to, right after a meal, it's going to allow for glucose to spread out all across these different organs, okay? And I'm sure you guys have heard that liver is the organ responsible for maintaining your blood glucose levels, right? Why? Because it can go through a process called gluconeogenesis, meaning I'm making glucose from non-carbohydrate sources. So for example, I can take the amino acid alanine. I can take this keto acid, lactic acid, or lactate. I can take glycerol. And using all of these, I can go through gluconeogenesis and make glucose, okay? Now the liver, what it likes to do is it takes the glucose that it makes from gluconeogenesis and stores it as glycogen, okay? And then whenever it wants to maintain blood glucose levels, it breaks that glycogen that it made from the gluconeogenesis, okay, and puts it out into the blood to maintain normal glucose blood concentrations, which is at 5 millimoles per liter, okay? So what does this mean? This means that the glucose, the dietary glucose, is not being turned into glycogen in the liver a lot, okay? In fact, the glucose that's coming from gluconeogenesis is being turned a lot into glycogen, and if needed, it will be broken down through a process called glycogenolysis into glucose and then put out into the blood, okay? And the making of glycogen is also known as glycogenesis, okay? And this is actually stimulated by the hormone insulin, okay? Stimulated by the hormone insulin. Okay, now let's follow all of these different organs and what they do with it, okay? So first of all, let's go to the pancreas. The pancreas is also going to have this GLUT2 transporter, okay? GLUT2 transporter, which again, if you remember, I said it has a high Km. So it's going to take in glucose, and that glucose is going to stimulate the beta cells of the pancreas. You know the beta cells of the pancreas, they make a specific hormone. It's called insulin. And then that insulin will go to the liver and stimulate glycogenesis, right? And it would stimulate the removal of the apical membrane GLUT2 transporter, okay? Cool. What else? What else does this insulin do? Well, you know this insulin comes over to the adipocyte, okay? This insulin comes over to the adipocyte and stimulates the recruitment of a specific type of GLUT transporter called GLUT4, okay? It also does the same thing for the skeletal muscle, okay? It recruits this GLUT4 transporter from specific storage vesicles, okay? They're called GLUT4 storage vesicles, and they're placed inside of the muscle cell. Insulin stimulates it to come and be placed on the membrane, okay? So insulin brings these GLUT4 channels here right after a meal, right? These GLUT4 channels will migrate towards the membrane and they'll sit there waiting to absorb glucose, okay? Now you might be thinking, okay, well, how do these cells get glucose in the absence of insulin, right? Because they have GLUT4, right? But how do they get glucose in the absence of insulin? Well, they get it mainly through this GLUT1, okay? So glucose can also go through this GLUT1 that's present in the adipocytes and the skeletal muscle, okay? But majority of the glucose after eating is actually cleared through these GLUT4 transporters that are insulin stimulated, okay? Cool. So what's going to happen to glucose in the adipocyte? So you know this glucose, it'll come in, okay? And again, through a series of enzymatic steps, I can actually turn this glucose into something known as glycerol 3-phosphate, okay? And you know an adipocyte has a lot of fat in it. It has fatty acids. I can combine that glycerol 3-phosphate that I just made from glucose and the fatty acids, and I can make a triacylglycerol, okay? That's a way of storing fat. So you know what I just did? I just turned my glucose into fat, okay? So that's one thing that can happen. At the level of the skeletal muscle, okay, I can get that glucose, okay? And that glucose can either go be used for energy. So for example, if we really need it for energy, like for working out, it's gonna go again turn into some pyruvate, right? And then go to the TCA cycle and then to the electron transport chain. However, this glucose can actually be turned into glycogen, okay, for storage and then for use later, right? So let's say I'm hitting a workout in the afternoon after I just ate. Well, that glucose got turned into glycogen, and I'm going to use that glycogen that I stored when I'm working out in the afternoon. So what I'm going to do is, after I stored that glycogen, when I'm working out, I'm going to break that glycogen into glucose, let it go through, the electron transport chain and make energy, okay? So that's the kind of fate of glucose in all these different cells. And in fact, 85% of insulin stimulated glucose uptake is done by the skeletal muscle, okay? And about 5% of the insulin stimulated, actually, I'll just do the same as last time. Stimulated glucose uptake is done by the adipocyte, okay? So when I mention insulin-stimulated glucose uptake, I'm talking about this GLUT4, okay? So after I eat, insulin gets released, right? I eat glucose, insulin gets released. We saw that here. And guess what happens? That insulin will then go and recruit GLUT4, okay? And 85% of the glucose that gets uptaken in an insulin-dependent manner is done by skeletal muscle, and the other 5% is done by the adipocytes, okay? We'll talk more about how much glucose is uptaken and whatever when we talk about glucose homeostasis in another video, okay? And then the last thing I want to discuss here is that glucose can actually go into the brain as well, okay? You know the brain, it only uses glucose, okay? It can only use glucose because of this thing. The presence of this thing called the blood-brain barrier. The blood-brain barrier is very specific in what it lets into the brain. So for example, lipids cannot come in, okay? They're too large, they can't go in. So the brain mainly uses glucose as its preferred source of fuel, okay? So here's that glucose. Now, in the blood-brain barrier, there's going to be a GLUT1 transporter, and the glucose will go through there. And it makes its way into the brain. Once in the brain, in order to get into the neurons that are in the brain, it has to go through a specific transporter called GLUT3. GLUT3. Okay, and then the glucose will then be used for an energy source here. Okay? As a matter of fact, the neurons in the brain actually create very little lactate, and they have a high aerobic capacity. Okay? So they make very little lactate, but they make a lot of CO2. Okay? Okay, so that's how the brain gets its energy. Now, also do notice that in prolonged starvation states, the brain can start using ketones as a source of energy, but again, its more preferred source is glucose, okay? Now, a little bit about the KMs of GLUT1 and GLUT3. So GLUT1 has a low KM, okay, meaning that it has a high affinity for glucose, but a low capacity for it. But the Km of GLUT3 is actually lower than GLUT1. So this is important. This is important because if GLUT1 has a higher Km than GLUT3, right? It relies more on the number of glucose than the affinity for it, okay? Which means what? Which means that a low glucose concentration could actually limit the amount of glucose getting into the brain. However, it has been shown that it is not until the blood glucose concentration falls less than 2 millimoles per liter that brain function becomes impaired, okay? So even at less than 5, all the way to 2, the brain function remains fine, okay? So that's just that little bit of tidbit, okay? Now, that's going to cover everything in relation to carbohydrate digestion and absorption. One last thing here is this little flow chart I made with regards to the digestion. So with all the enzymes on the brush border. So again, remember that lactose uses the enzyme lactase, and that gives us glucose and galactose. Trehalose uses trehalase, and that gives us glucose and glucose. The complex of sucrase and isomaltase. Isomaltase, remember I said, is the only enzyme that can cut the alpha-1,6 bonds. So it cuts isomaltose and the limit dextrins. And that gives us glucose, okay? Sucrase is able to cut sucrose, which gives us fructose and glucose. But sucrase can also take on things like maltose and maltotriose, okay? Finally, maltase can cut maltose and maltotriose, which both give us a whole bunch of glucose, okay? Cool. Now, that's going to finish carbohydrate digestion and absorption. Again, if you guys enjoyed it, please go ahead and subscribe. I'm going to have more contents on this. We're going to talk about glucose homeostasis. We're going to talk about lipid digestion, protein digestion, all of that coming soon. Okay? And I'll see you next time.